I'm Eva,
And I'm Naomi.
and this is our video on protein synthesis. Protein synthesis is the process of building proteins. It occurs in two steps, transcription and translation.
The first step is transcription. Transcription occurs in the nucleus of the eukaryotic cell. The initiation site is DNA and RNA polymerase and binds to the DNA and separates the strands. RNA polymerase uses one of the DNA as a template strand, elongating the DNA strand. Then it assembles the nucleotides along the strand of RNA. The mRNA nucleotides are adenine, uracil, cytosine, and guanine. RNA polymerase will then get the termination signal and stop transcribing the DNA. When finished, the direction of the transcription is 5:3, and you have an RNA transcript complementary to the DNA strand. The mRNA strand then undergoes mRNA processing, which is the editing of the RNA strand. Intron pieces not expressed in the proteins are recognized and spliced out, leaving only extrons and express code proteins. The group at the 5 end is called the cap, while the group at the 3 end is called the tail. The mRNA is now a genetic code for making proteins and leaves the nuclear membrane in order to find the ribosomes.
Once it finds a ribosome, it then enters the cytoplasm, where the second step of protein synthesis occurs, translation. Translation occurs in the cytoplasm of the cell. As mRNA finds a ribosome, rRNA, the small subunits then bind and read the mRNA. As mRNA elongates, the A site comes in, accepting tRNA bound with amino acids. The large subunit takes care of production of the peptide bonds once they are formed. This is what happens, and then it would connect like this, like so. mRNA codons are shown. The tRNA anticodons attach themselves according to complementary base pairing. So guanine would connect to cytosine, and adenine would connect to uracil. And the P site holds the tRNA that carries the amino acids. Most of the time, the start codon is AUG, but this could vary from time to time. After each tRNA connects itself to the complementary mRNA, they leave. However, they leave behind an amino acid, so the peptide chain would just slowly get removed. These amino acids keep forming along the mRNA strand, bound together by a polypeptide chain, which disappears in the E site, or the exit, and there is a stop codon informing the tRNA to stop forming the polypeptides. And when the finished chain, the polypeptide folds into an active protein, and it continues to perform its functions in the cell, leaving free amino acids that are complementary due to the polypeptides and the mRNA, and that is protein synthesis.